describe the amazing work today. So my privilege to introduce Lauren Jackson. I'm Matt Johnson, professor here in the wildlife department. And Lauren has been an amazing addition to the lab. So I think, as a lot of you know, Lauren is the latest, I guess, in a growing line of students that have studied barn owls, and every student brings some unique aspects to the project. And that's certainly the case for Lauren. Can you go forward one? Hold on. There you go.
Okay.
And I love to introduce students with a quote. And the quote I've chosen for Lauren is from Shakespeare, and from Hamlet. And he said, "To thine own self be true." And I think that's true for Lauren herself. She brought in all this energy and all this excitement about studying behavior in ways that my lab hadn't previously talked about or explored very much. And as you'll see from the research, she's also really interested in the individuality of barn owls and how that affects how they live in the world. So Lauren's project really focused in a new area for my lab in the Central Valley of California, Lodi. And Lauren did an amazing job reaching out to all the farmers and facilitating a really vibrant community of folks interested in this research. And now she has some results to share with us. So I can't wait to see and see how she's put it all together. So take it away, Lauren.
Okay, thank you so much, Matt, for the intro. Thank you, everyone who has joined on Zoom. I'm sorry for the technical difficulties, but you can just imagine all of our smiling faces here in the room. So let's go ahead and get started. I'm super excited to talk about my work today entitled Evidence of Personality and Movement Syndromes in a Mobile Predator, and how individual hunting strategies shape ecosystem service potential by barn owls in an agroecosystem. So a concept that most of us are familiar with likely is the widespread ecosystem services that animals across several taxa provide to landscapes throughout the world. And most research on this topic has really focused on the economic valuation of these services, as well as identifying key groups for targeted management. But these services are also often described at the level of species, populations, or communities. However, ecosystem services are constantly being delivered through the behavior of individual animals themselves within these larger groups. And the fact is, the presence of these service-providing species alone may not always equate to an even distribution of services across landscapes. In reality, these services really do depend on how individual animals do their day-to-day lives and where they're doing everything and how consistently they actually perform these service-related behaviors. And so this idea of individual variation among animals has become increasingly recognized as an important driver in lots of ecological dynamics. And several studies at this point have identified that animals within the same populations can vary quite substantially in activity levels, movement strategies, foraging behavior, and several other behavioral domains. And a lot of literature has also found that in many cases, by pooling individuals in analyses, we run the risk of obscuring meaningful differences and ecological trends that appear at the individual level. So one useful framework that has started to gain popularity for evaluating stable behavioral differences among animals is animal personality. And in an ecological context, personality is typically defined as the behavioral or physiological differences among individuals of the same species across time and context. And personality has been characterized through a lot of different distinct axes, including neophobia, aggression, exploration, just to name a few. And in many cases, these axes can also co-vary with one another to form broader behavioral syndromes, often referred to under the paradigm called the Bolshoi continuum. And this is often used to describe the temperaments of animals kind of on this continual sliding scale. And while several studies have identified clear differences in behaviors across taxa, many more have also narrowed in on the distinct repeatable differences in movement strategies or space uses among individuals. And so this typically emerges in differences in foraging behavior, ranging, site fidelity, just to name a few. And these differences can ultimately alter the spatial distribution of ecological interactions and ultimately influences where animals forage, which habitats they exploit, and which resources they ultimately encounter. And this is really particularly relevant to mobile predators, which offer a useful system for examining links between individual variation and ecosystem service delivery. Because predators, while they're constantly making decisions about where they should go, which habitats they should hunt in, which prey to exploit, their decisions affect not only their own behavior, but also that of their prey. And so these individual decisions at the upper trophic levels can ultimately cascade down into subsequent levels and affect the prey behavior through both perceived predation risk as well as direct consumptive risk. Barn owls are one mobile predator that offers a really strong model system to test some of these relationships between individual variation and ecosystem service delivery. They are widely used at this point as biological control agents for several rodent pests in agricultural systems throughout the world, including right here in California, especially in California's winegrape vineyards. And these owls have several life history traits that make them really well suited for pest control. They are broadly distributed throughout the globe. They are highly mobile. They have a very high rate of prey consumption, particularly during their breeding season. And they will readily use human-made nesting structures, which allows them to exist in a relatively high density on local agricultural landscapes. And so this accumulation of characteristics allows them to exert this top-down force on rodent populations in these systems. And because of these qualities, farmers have begun adopting this barn owl nest box system type program to encourage barn owls to occupy their landscape and provide rodent control to their surrounding areas. And so by installing these boxes, these nests will provide nesting habitat for barn owls during a critical time period of their life, and owls can in turn provide some rodent control to these surrounding landscapes. And this tends to work pretty well because barn owl diet is composed primarily of small mammals, especially rodents such as gophers, voles, and mice that tend to be really prolific pests in a lot of agricultural systems, especially California's winegrape systems. And while a lot of research has gone into understanding barn owls as agents of pest control, most of these studies, like Matt mentioned, have focused on evaluating the suppression benefits at the population level. But this implicitly assumes that individual owls will contribute similarly to pest management, when in reality, these services may vary depending on the characteristics of the individual owls themselves. And beyond potential behavioral variation, these owls are also known to exhibit distinct polymorphism in their physical plumage traits. So these owls tend to vary across the spectrum of red to pale and spotty to no spots at all. And these traits can be attributed to two main pigments known as pheomelanin and eumelanin. And several studies on barn owls to date, especially on the Western barn owl, our European counterparts, have linked differences in polymorphism to behavioral and physiological differences as well, with ultimate effects on things like habitat selection and movement strategies. And so while this hasn't been studied as extensively in the American barn owl quite yet, the plumage variation here offers another unique opportunity to test if visible phenotypic traits can be linked to ultimate behaviors that influence ecosystem function. And so together, these concepts suggest a pathway by which individual traits can ultimately shape and influence ecosystem service delivery and how animals interact with the landscape. And so here we're hypothesizing that plumage and behavior are associated with variations in movement and hunting behaviors, resulting in differences among owls and their use in vineyard landscapes for foraging, and consequently, the potential contributions to rodent removal on vineyard landscapes. So this is essentially linking how do individuals vary and what does that mean for their impact on ecosystem services in these systems. So to begin addressing these objectives, to begin addressing this hypothesis, we formulated 4 primary objectives for this project. The first of which is that we're aiming to determine if behavioral or movement traits are repeatable within individuals, indicating consistent individual differences and therefore potential axes of personality. Next, we aim to evaluate if repeatable behavior and movement traits covary to form any sort of broader personality syndrome, such as described in the gold-shy continuum. And we also want to test relationships between plumage traits, behavioral variation, and see if plumage predicts behavior, or if plumage and behavior are associated with movement or hunting strategies. Finally, we will be building towards understanding if plumage, behavior, and movement strategies can predict hunting use and selection in vineyards. And so to test these objectives, we headed out to the field. Our study site for this project was located around California's Central Valley, around the city of Lodi. And this area is really characterized by a hot summer Mediterranean climate, which makes it perfect for growing wine grapes. And Lodi and the surrounding areas are really dominated by this wine grape production, but also interspersed with other crop types, including fallow fields, developed areas, and riparian and woodland habitats. And the farmers in this region have been using barn owls as a method of pest control for several years in this area. We were able to use an existing network of nest boxes to test some of these questions. And this map to the left here shows a network of about 170 nests that we monitored for breeding occupancy in 2025. And so using these boxes, we were able to successfully capture 42 adult male breeding barn owls during the spring of 2025. And so here we trapped each owl using a walk-in trap device at the nest during their chick rearing phase. And we typically target the chick rearing phase in order to take advantage of the high parental motivation at this stage, as well as minimize any risk of this nest abandonment. And we further decided to target only male owls for this project specifically because males have been found to exhibit the bulk of the hunting behavior among the pair, and we really wanted to maximize how much hunting data we could collect to answer some of these questions. So once each owl was captured, we recorded data about their sex, age, and morphometrics as well. We also collected information about the plumage characteristics of each owl by taking plumage photographs using the ventral surface of the breast, and by simply placing these owls on a standardized surface and using a customized white balance image photo setting for each photograph taken. Finally, each owl was then fitted with a GPS-accelerometer tag that it wore for approximately a week, during which time the tags collected 1 GPS point per second and 50 accelerometer readings per second, and were active during the first 6 hours of each night. And at the end of each deployment, we would retrieve the owl, simply remove the tag, and be able to download all of that data. And so also while this GPS tag is deployed and collecting GPS data and the owl's out flying around collecting data for us, we conducted a behavioral assay known as a novel object test. And this is one popular method in animal ecology studies that is used to evaluate individual boldness by measuring neophobia, which is essentially fear or avoidance of a novel stimuli. And the way this test works is really simple. We simply place a novel object on top of the nest and go ahead and measure each owl's response to that object. And so this works well in this system because breeding owls are very highly motivated to return to the nest in order to provision their chicks on every night. And so they must approach this object in order to actually access their nest. So for this test, we designed 2 distinct novel objects, each of which varied in shape, color, and texture in order to account for any effect of any one of these. And we're also designed to be as visually detectable as possible under low light conditions by barn owls that are encountering these stimuli in the dark. And so we used an alternating control treatment schedule as well, in which each test was buffered by a control night. And we randomized the order of the object exposures evenly among owls in the system too, to control for any unexpected effects of treatment order. So while this GPS deployment and novel object tests are going on, we collected continuous video data at the nest using nest-mounted cameras. And this data enabled us to take a closer look at owl behavior in response to the object, but also on nights when the object wasn't present at all. And so we use an ethogram approach here using the behavioral scoring software called Boris to extract a 90-second scoring window over the first visit of each night. And from these ethograms, we extracted counts regarding prey deliveries or the number of times the owl landed on the nest, for example. We also extracted latencies to first attend the nest or abort a visit to the nest. We determined the proportion of time each owl spent in the camera frame or doing continuous behaviors such as flying, perching, or entering inside the nest. And so now that we have all of this data back, we can talk a little bit about how we process all this information and the analytical workflow used. We did implement a lot of different streams of data here. It gets a little bit confusing to kind of take all in. So we're going to go through a quick flow chart just to show the process, and then we'll dive into deeper specifics of each step. I do also want to mention we're going to be jumping back and forth between methods and results through each section of this analysis. So don't get scared when we're still in methods towards the end of the talk. Okay, so here we are after coming back from the field. We have all of our nice plumage photos, nest video data, and our GPS data. And we start off by further deriving a set of candidate variables for each of these behavior and movement data streams to divide behavior into baseline and neophobia responses as well. These variables will get entered into a repeatability analysis to identify any repeatability and traits that may serve as potential axes of personality. And we'll further filter these down to the only repeatable traits and owl level traits for further analysis. So at this point, we will have plumage, behavior, and movement data kind of narrowed down a bit. We'll go ahead and then loop in some habitat availability, use, and selection data. And all of these variables together will get entered into a single final analysis workflow to test associations among each of these distinct domains. And also test a few different model sets to predict, to associate with, oh my goodness, to test the associations between each of these individual level traits and their ultimate influence on ecosystem service delivery in this landscape. So I really want to highlight that these are the meat of this analysis and that all of these steps building to these final questions will help us answer some uncertainty about the link to ecosystem service delivery. Okay, so let's start off with our simplest analysis among these. This is our plumage data. And recall we started with photographs of the ventral surface of each of the owls here, and then we went ahead and just cropped these down to a 40 by 60 millimeter region from the upper center part of the breast, where we tend to see the most color variation among individuals. We then further separate these photo samples into spotted and non-spotted pixels for the respective pigment analyses. Redness would be calculated from the non-spotted pixels, so all those white feathers, by looking at the red-green channel intensity ratio. And spottiness will be calculated as the percent of pixels in the photo that are spotted relative to non-spotted pixels. And so let's take a look at the GPS and ethogram data here as well. Using the GPS tags, we first extracted two metrics that were most relevant to nest approach and near nest behavior. The first one we derived here, we call nest contact latency. So this is going to describe the delay between an owl's first entry within 25 meters of the nest and the first time the owl physically touches the nest box. We were able to cross-reference our video data here as well to derive precise times for these variables. And approach latency in general is a common metric that's used in novel object tests to describe an animal's willingness to approach this type of novel stimulus. Secondly, we're going to be using this GPS data as well to determine the proportion of time an individual spends within 25 meters of the nest. This also reflects kind of the differences in proximity behavior and how willing the owl is to be near the nest with and without that object presence. So these two variables will kind of reflect each owl's response to novelty. Next, we'll take a closer look at the ethogram-derived data that we extracted from the video footage for each owl. And first, we will be taking a look at several continuous variables, including how long the owl spends flying, perching, or attending the nest by feeding chicks or entering the nest. We will also be looking at the counts, so number of times each owls land on the nest, abort a visit, or complete a prey delivery successfully. We'll also record if an owl was carrying prey or not during their visit. And finally, derive several latencies for the delay to first land on the nest or delay to first attend the nest. So again, feeding chicks or entering the nest. And so using these two combined with GPS and ethogram data together, we ultimately derived a set of 11 nightly variables that we felt were most relevant to the questions being asked, and then go ahead and further filter this data into two subsequent categories. So our baseline category will be reflecting control nights only. This represents an owl's assumed standard behavior in absence of a novel object, whereas the delta neophobia response will represent each owl's response to the novel object by comparing treatment and control nights and evaluating how this response changes between the two. And so for each of these distinct categories, we derived 11 variables, which will be assessed in future analysis. So for now, let's take a quick minute to talk about how we handled some of the movement data in the system. We successfully collected GPS data from 42 owls over 297 nights, representing approximately 6 million GPS fixes. So these owls were working hard to collect us lots of good data to work with. And the first thing we did here was go ahead and filter this data down to the precise deployment periods, further clean it up by remove any missing or low quality fixes, and then resample everything to 10-second intervals. We then fitted a hidden Markov model to derive 4 distinct latent states here, which included perching, commuting, and 2 types of hunting behavior, and then further collapsed this to hunting behaviors only as a single category. And using these data together, along with the full nightly metrics, we were able to calculate a suite of owl-level nightly metrics to describe space use and movement strategies. And so here we initially selected 12 movement traits to represent a few different complementary and biologically interpretable dimensions of hunting strategy or space use. And among these, we determined how far owls traveled each night, how much area they actually used for hunting, the diversity of land cover types they used for hunting, as well as site revisitation and overall path tortuosity. And so with these variables assembled across each data frame, we were left with 11 baseline variables, 11 neophobia response variables, and 12 movement variables. So here's where we're at now. We have our slightly narrowed down set of variables, and we're going to go ahead and proceed with a repeatability screening. And so repeatability screening is a really critical step in evaluating animal personality, and this is typically used to determine the proportion of variance that you observe in a trait that is attributable to among owl differences. And so here we calculated repeatability using an interclass correlation coefficient from linear mixed effects models that we fit to each candidate variable. And so models were fit to repeated observation with owl ID as a random intercept and included multiple fixed effects for the date, so time of season, treatment order, as well as nestling information and other weather or environmental covariates. And so here we consider traits that were, to be biologically meaningful candidates for subsequent analysis and therefore repeatable when their ICC was reported to be greater than or equal to 0.3. And this value represents a typical modest value that's reported across several animal personality studies. And for any repeatable traits that we did retain, we then extracted model-adjusted estimates to use in our final trait assembly. And so this brings us to our first exciting result of the study. We found that barn owls in this population did exhibit repeatable variation in terms of multiple behavioral and movement traits. And so from our baseline variables first, we ended up retaining 3 distinct traits, including nest contact latency, nest area residence, and proportion of time in the camera frame. From our neophobia response, we retained only 1 trait. This was the delta latency to attend the nest. And from all 12 movement traits, we saw repeatability, and we ended up selecting 6 that we felt were the most biologically relevant to the questions we were asking for further assessment. So we'll talk a little bit more about these in a bit. But first, here's a quick plot summarizing the findings among these repeatable variables. This panel on the left here shows the selected traits and their repeatability estimates and the 95% confidence intervals plotted on those horizontal lines. And that white spotted dot you can see going vertical is our ICC threshold set at 0.3. And the traits here are just colored by domain grouping just to keep things a little bit more organized. But when we look at this plot on the right here, we can see that the spread of the raw data is really highlighting how much variation can exist between all these different individuals, yet within individuals, we are seeing some degree of consistency within each of these traits. And so just to summarize our first result here, we can confirm that several traits measured in this population were repeatable among these owls, and these can potentially represent axes of personality, but we do observe that there is some repeatability here. So super exciting, and this lets us move forward with some next steps of our analyses. All right, so now that we've identified all these traits, we want to test for any behavioral syndromes or the possibility to reduce these traits within domains into fewer axes. And so while we look at the correlation of these traits, we can determine where there may be redundancy and potential to derive simpler composite axes of behavior. And so we didn't find any high repeatability among the behavioral traits, correlations among the behavioral traits, surprisingly, but we did find that many of the movement traits were highly correlated with one another. And so because of this, we went ahead with a PCA to derive composite axes of movement and ended up plugging in those 6 variables that we determined to be the most relevant for this system. So these included ranging extent, hunting habitat diversity, site revisitation, core area use, as well as bout duration and path tortuosity. And so together, this PCA effectively simplified these movement traits into 2 main axes that described space use and hunting search strategy. And together, these axes described about 65% of the variance, with PC1 looking a bit like a space use strategy. And this first axis here was mainly loaded with hunting for area, hunting site revisitation, and ranging extent. Meanwhile, PC2, we termed the hunting search strategy, was mainly loaded with path tortuosity, hunting habitat diversity, and hunting-bout duration. And so to get a better idea of how these look in reality, let's take a closer look at some of the owls from the opposite ends of these PC extremes. So looking here first at the PC1 axis, this is what we call our space use strategy. We see that owls with a low PC1 score generally correspond to paths with larger, farther, and less heavily used hunting areas. So this owl here clearly ranges a bit farther from the nest, explores a greater variety of hunting areas, and more superficially. Whereas an owl with a higher PC1 score will exhibit smaller, closer, and more heavily used hunting areas. So this owl here tends to stick closer to the nest and repeatedly use the same location. Meanwhile, for our PC2 axis, this is the one we termed hunting search strategy. We see that owls with a lower PC2 score generally correspond to more directed, persistent hunting over less diverse landscape and with longer hunting bouts. Whereas an owl with a higher PC2 score generally has paths that are more tortuous, cover more diverse landscape, and over shorter bouts. So they tend to divide their hunting into shorter, continuous paths of time. And so these two kind of compare and contrast against each other, and you can see a lot of variation happening here among these individuals in terms of what's going on with their space use and overall movement strategies. And so this kind of reflects our next exciting result from this data set, and tells us that we can identify a few movement syndromes that summarize these traits effectively from our greater set of movement variables. And so let's just take a quick refresher of where we're at now. We have assembled our final traits for plumage, behavior, and movement. And so now we can go ahead and loop in our our habitat and landscape metrics. And so let's go ahead and dive into how we derived plant cover. For this data set, we used a combination of two different data sources. These included the 2025 Cropscape data as well as the 2025 Vineyard Mapping Project data. And from these, we were able to assemble a 10-meter raster that we further collapsed into 7 distinct land cover types. So these included vineyard, pasture and grass, orchard, row crop, as well as fallow and removed, woodland and riparian, and generally avoided classes like urban, developed, and water that we collapsed into a single group because owls don't tend to use these too much. Now, using this data, we extracted information about the availability of landscapes around the nest, including the proportion of vineyard habitats within 1,000 meters of each owl's nest box. So this serves as a baseline availability for vineyard for each owl. We also extracted the proportion of hunting steps on vineyard habitat as a metric for vineyard hunting use. And finally, we determined the actual vineyard hunting selection of each owl using an integrated step selection analysis. And so to talk a little bit more about this selection step, we fitted a mixed effects ISSA using a Poisson trick here to evaluate each owl's specific selection for vineyard land cover. And so here we started with one use step and generated 15 available steps for each of those use steps. And in this model, we included several fixed effects, including movement kernels, endpoint land cover to derive selection, as well as an interaction between start point land cover and each of these movement kernels. So this effectively allows this allows movement to vary by land cover type and reflect kind of a natural tendency for owls to potentially move differently across different habitat types. And so here we also included random slopes for land cover selection, and we derived individual vineyard selection estimates relative to our reference level, pasture, grass. So all of that to say, we determined each owl's individual selection for vineyard land cover. And so here, across these different landscape metrics that we looked at, we found that owls tended to vary really widely in the proportion of hunting on vineyard habitat, anywhere from 1 to 98%. So this means that in the pink here, you can see our vineyard hunting use. that owls spent almost anywhere from no time hunting on vineyard habitat to almost exclusively hunting on vineyard. So that's super interesting to see. There's also this kind of clean cascading effect here where everyone seems to be doing something a little different. And next we looked at the general baseline availability of vineyard around each owl's nest. And this tended to range pretty broadly as well, anywhere from 9 to 80%. And finally, we also observed a lot of variation in individual level vineyard selection, with some owls selecting vineyard much more strongly relative to pasture and grass, and others selecting vineyard much less. However, when we look at this data as a group, we see that owls in their selection tendencies across this population select vineyard very similarly to pasture and grass, and this is really to be expected. We generally see that owls reflect these types of tendencies across land cover selection analyses in the literature, but this group seems to be avoiding most other land cover classes, not extremely strongly in many cases, but one thing I do want to point out is the apparent variation in the individual level selection across several of these groupings. So we see a big point spread for vineyard as well as fallow and removed and a bit over orchard. So each of these owls tends to have a little bit different selection preference, and this point spread reflects a lot of that variation among the population that we sampled. And so here's where we're at now. We finally reached the bottom of this cascade where we have all of our final traits for plumage, behavior, movement, and now habitat metrics. So let's go ahead and get started with our final analyses to take a look at how these variables all relate to one another. So here we fit a set of linear models to examine associations between plumage, behavior, movement, and vineyard use and selection. And so for this final analysis, we ended up using 41 owls and excluded one owl due to lack of data across several nights, as well as a novel object test that failed. And ultimately, we used an AICC model selection to compare these candidate model fits. So starting off, we want to understand the relationship between behavior and plumage. So how do plumage traits potentially predict behavior? And what we found in all cases is that the null model came out on top here, which interestingly revealed little support for plumage as a predictor of any of the behavioral traits that we tested here. Now looking at the model set for movements being predicted by plumage and behavior, again, in both cases, we saw that the top-ranked model was null for each of these once again. So the predictive effect of plumage or behavior on either of these derived PCA axes seems to be relatively weak. And so, again, a little bit surprising, but we're seeing a bit of a consistent trend here that overall these traits seem to reflect some sort of independent domains among them. All right, so for our final set of models, we can examine the relationship between our really interesting response variables, which are going to be the vineyard hunting use and vineyard selection variables. This is going to be the model set that links each of these individual level domains and traits to the ultimate ecosystem service potential. So we're asking here, how does plumage and behavior and movement potentially influence each owl's vineyard hunting use or vineyard hunting selection? And in these models, we found some interesting results. So I do want to mention, we also included vineyard availability across each of the models as a baseline control for effects of vineyard habitat near the nest. But here we found that the top-ranked models included some of our different domains, and especially we saw that across both of these, movement strategies were included as a predictor. We also saw that for vineyard hunting use, neophobia appears in the top model, which is pretty interesting. But let's go ahead and take a little closer look at each of these model estimates. So for the summaries of each of these models, we see that in the top-ranked vineyard models, among vineyard availability and hunting search strategies are both really coming through as strong predictors, seem to have some sort of association with vineyard hunting use and vineyard selection in this system. And the other traits that are included don't seem to have as strong of a signal. So the beta estimates here are essentially revealing that owls with a greater vineyard hunting use or selection typically have these lower PC2 scores. And so let's take a little bit closer look at what that actually looks like in terms of a relationship here. Again, visualizing these model summary outputs, we can confirm that the higher an owl has vineyard use and selection, the lower its movement PC score tends to be. I also just want to mention the point size here represents vineyard availability, but kind of a baseline thing, so we don't want to focus on that too heavily right now. And to make this a little bit more comparable, let's take a second look at those GPS tracks that we looked at earlier from each ends of these extremes. And so first, this owl on the left side of the plot typically exhibits a really high level of vineyard hunting use as well as vineyard hunting selection. And this corresponds to a generally lower PC2 score. And this is exhibited across this owl's track by a more directed, persistent hunting strategy with less diverse land covers used and longer hunting bouts. Meanwhile, if we look at the owl with the points on the far right of these plots, we can see that he selects vineyard less and uses vineyard less, but he has this higher PC2 score, which corresponds to more tortuous paths, more diverse land covers used, and a shorter habitat bout tendency. So these two differences in movement strategy seem to reflect onto the relative amount that owls are selecting or using vineyard in these landscapes. And so just to summarize this result, we find that these associations between movement strategies and vineyard use or selection can be pretty interesting. But let's talk a little bit more about the overall findings from these results. So just to relate back to our initial objectives, we first aimed to determine if behavioral or movement traits were repeatable and therefore indicated consistent differences among individuals. And here we found that yes, several of the behavior and movement traits that we assessed exhibited some level of repeatability, and that these consistent individual differences may act as potential axes of personality. Next, we aim to identify behavioral or movement syndromes among these domains, and here we found partial support with no real behavioral syndrome appearing, but distinct movement syndromes appearing out of PC1 and PC2. Next, we wanted to test these associations between plumage, behavior, and movement strategies, and here what we found is that, interestingly, there was little association among any of these domains, with each of these axes appearing to act almost independently of one another in this system and among these samples. And finally, we aim to determine if plumage, behavior, or movement strategies predicted vineyard hunting use and selection. And again, here we found partial support with our movement strategies coming through strongly as being associated with vineyard hunting use and selection, as well as neophobia appearing in our vineyard hunting use models. So this is really the core analysis that we wanted to answer questions about, is what is the association among all these traits and this ultimate ecosystem service function? And we're seeing some relationship here between movement and vineyard habitat use and selection. And so what does this all mean together? First off, the fact that these traits were repeatable among individuals suggests that barn owls may have distinct axes of personality that emerge across different contexts. Interestingly, though, we didn't find any clear evidence of this broader personality syndrome idea among plumage, behavior, or movement strategies, because these domains did not clearly covary with one another. So this could be explained by the fact that these traits might be operating at different spatial scales and under different motivational contexts. So during breeding, the male owls are really strongly pressured to feed their nestlings and attend to the nest. And this motivation might essentially constrain behavior enough that any subtle differences among plumage traits, such as redness or spottiness, could be effectively swamped by nestling demand, prey availability, or mate behavior. Not only that, though, we also measured neophobia at the nest in a really localized setting, and so behavior here might be heavily shaped by the nest box environment and also pressure to provision the nestlings. And an owl's sensitivity to novelty might just be higher near their immediate nest environment than across their full nightly range. And so we didn't observe a lot of differences here associations here between neophobia and other traits, but we did see that nightly movement and hunting strategies might be shaped more strongly by this broader local landscape configuration, prey availability, and potentially the spatial arrangement of foraging opportunities. So rather than reflecting one greater personality syndrome here, it looks like we don't see the bold shy continuum quite as we expected, but these traits that we captured might reflect distinct axes of personality that just associate differently than we expected. And so what we're seeing in this study is that key thing here is the movement axes may be most directly relevant to ecosystem service delivery rather than some of the traditional behavioral axes alone, such as neophobia. And so at the same time, individual behavior does not operate in isolation. Landscape context still really mattered. Vineyard availability near the nest was a really important predictor of both vineyard hunting use and selection. And so the broader point here is that neither nest box occupancy alone or vineyard availability can guarantee a uniform pest control potential, but these predator-mediated ecosystem services still depend a lot on the interaction between landscape opportunity and the spatial strategies of individual predators. So it's not just whether barn owls are present on the landscape or if vineyard is present near the owls, but also how owls move and the ultimate realized locations of where they hunt. And so as with any ecological study, this one doesn't come without a few limitations of its own. I will say quantifying personality in animals is generally difficult, and doing so in the wild is also really tricky. So we definitely had to compromise between a few constraining factors in this study. The first of which was our obviously short sampling window and sampling of male barn owls, which gives us an overall sort of limited context about the repeatability of these traits across broader time, seasons, or contexts. And so here we also use proxies for rodent removal, so we didn't quite test the direct ecosystem service delivery here in terms of successful hunting or direct rodent counts, but rather looked at this pest control potential by owls in this system. And one important thing to notice as well is that we weren't able to make any really definitive conclusions about causations of the patterns in this system. So without any additional data, we can't quite yet separate the intrinsic movement tendencies of individuals from their potential responses to the local landscape. And so this sort of introduces questions about if individual owls are choosing to hunt a certain way and therefore encountering different landscapes, or if the local landscape is then shaping the subsequent movement strategies that we observed. And so regardless of this unknown that remains, these realized movement strategies that we observed still have meaningful consequences for the downstream ecosystem service potential in this system. And this leaves a lot of room for future studies to address some of these shortcomings. Future work could really focus on implementing a longer-term and multi-seasonal sampling design, especially across a variety, sort of a gradient of environmental conditions and prey availability as well. And so tracking individuals across years, nesting sites, movement strategies, these things could really help clarify the mechanisms of causation in this system between individual tendencies, habitat selection, and ultimately pest control. Finally, adding a more direct quantification of rodent removal, like strike locations or the success or failure of strike attempts, where these occur, as well as rodent abundance on the landscape, could help elucidate the more direct implications of ecosystem service delivery in this system. And happy to report we are currently working on this with Tim Bean and his students down at SLO. So stay tuned. We might have some more rodent news soon. Okay, so just to summarize, this study offers a really exciting and fun first look at the potential axes of personality in a nocturnal and mobile predator. And so this suggests that barn owl contributions to pest management depend not only on the nest box occupancy and vineyard availability, but also on the individual differences that we can observe in movement behavior. And recognizing this type of individual variation might improve our overall ecological understanding of how and why predator-mediated ecosystem services tend to vary across these types of landscapes. Another kind of broader note is that personality diversity has potential to buffer ecosystem service delivery under a variety of conditions and across time and context. And so we see that in general, personality diversity can help stabilize these types of rodent suppression services across all sorts of different years. If there's a tough year, a dry year, a hot year, some individuals may thrive in different conditions than others, and provides a more temporally uniform pest suppression across time. So this type of behavioral diversity is important to understand better, to get a better idea of this service and how this could change over the longer term. But with all of that, I would like to take just a quick moment to acknowledge all of the amazing and incredible people who made this project possible. First, I want to give a huge thank you to the vineyard owners, the land managers, the farmers and community members of Lodi, who not only granted me access to their lands, but also welcomed me into their community with open arms and such truly contagious enthusiasm. And a really special shout out to Stephanie Bolton, who was an integral part of rallying the community around this project. And if you know her, she's just a beam of light in a tough time for vineyard growers everywhere. So thank you to Stephanie especially. Next, I really want to thank my team of field technicians, Maddie and Noah, as well as other folks who helped get this project off the ground in the early days, especially Zoli Schneider. He helped me tackle almost every wrinkle at the beginning few weeks of this project, and honestly wouldn't have been as smooth without him. So thank you, Zoli. And of course, thank you to the Johnson Lab and my community here in Humboldt for supporting me through this process and for enduring truly endless hours of me talking about personality. I am sure you're all sick of hearing about it by now, but thank you. And a huge thank you to my parents and brother, who've just been my biggest cheerleaders for my entire life. They have just given me truly unwavering support and love through everything that I do. And I think that my brother might have even learned what a bird is for my benefit sometime recently. So thank you, Cole. I love you for that. And finally, thank you to my advisor, Matt, who has shared his love and passion of barn owls with me and has officially made me a owl-obsessed researcher myself. I'm just so grateful to be part of this project, and these owls have taken me around the world and back, and I'm so excited to see what's next. And thank you all for being here to listen to me today. I'm sure we'll have many more exciting conversations about personality in the future. So with that, thank you, and I'd be happy to take questions. Hey, Rob.
Wonderful, thank you. Beautiful presentation and amazing science. So especially, when it comes to assessing individual personality, like one of the things we hear about frequently is this sort of non-random subset of individuals that tend to colonize new habitats or be more amenable to things like urban or cosmopolitan habitats. And I, without knowing much about owls, certainly, do you think that the placement of these owl boxes captures from the full potential spectrum of behavioral syndromes in owls? Or do you think you have a subset of ones that tend to be sort of a priori more amenable to these relatively cosmopolitan habitats?
Yeah, that's a really great question. For anyone on Zoom who didn't hear, Rob's asking, do individual differences potentially manifest in an owl's willingness to use a nest box environment already? So we'd be sampling a biased portion of this population, essentially. And you're absolutely right. That is fully a possibility. We don't know at all about the effect of this, but it is possible that owls who are more willing to occupy nest boxes may be more tolerant of disturbance. Vineyards come with a lot of work and other activity that comes around them. So there's a lot of inherent disturbance and activity around a lot of these nest boxes. And so it's possible that some barn owls aren't using nest boxes at all. But yeah, it's a little tricky to know, and I wish that we could sample owls across the full distribution and catch them in trees and like the whole 9 yards. But yeah, that is definitely a point to note. There could be a motivation that drives this baseline occupancy of boxes too. Yeah, thank you. Great question. I see Christine is raising her hand on Zoom.
Yeah, and I actually also would want to see you. Is there any chance to see you?
Our video is not working in the room, so unfortunately.
That's a shame. That's a shame. Okay, so anyway, thanks very much. It was really exciting to listen to your study. That's amazingly complex to analyze also the data, and I tried to follow all your analysis, and there's one thing that I was not sure that I was kind of properly capturing, and that is how, well, first of all, I think it's amazing that you could bring down all these different measures of observations into something that you could measure if it's repeatable. Really, really congratulations for it. I mean, that's wow. Now, when you brought this down to these different levels of movement or hunting behavior, when you did the first evaluation of the markers that might be relevant, did you have a kind of retest reliability? So who was coding them, and was there another person coding them as well? And then would you agree on it, or would you put it into kind of an automated process?
Okay, yeah. Are you referring mainly to like the ethogram derived behavior scoring?
At the very beginning, yeah, before you put them into also a PCA, or when you try to say, oh, these are my 11 variables, and I was really, really attending to that from the recordings. So the question is if you feel that somebody else, like we would do it in Switzerland, would we be able to do the same thing? And then when you did it in your data at the different locations, is it only you who did the kind of that was this behavior or that behavior, or did you have two persons, or was it automated somehow now?
Okay, thank you. I think that's a great question. Thank you so much, Christine. To answer that as much as I can, we determined some of these traits that we wanted to test for repeatability kind of as we went, but also a lot of them were established a priori. So Matt and I especially did a lot of time where we were planning what could be the most relevant to measure in this system, and we knew going into the field even that we were going to be measuring this nest contact latency and the area or time spent within a certain area of the nest. And so a lot of our methods were directly pointed at trying to derive those variables. In terms of the other variables, such as the...
So can I quickly interrupt you? So if you did it and Matt did it, would you come up with the same solution that Matt, or were you sitting together?
I definitely would come up with something and then we talk about it and adjust a little bit. So our methods were mostly collaborative, I would say, and it's certainly subjective to some degree. So somebody else repeating the study with no knowledge of this information would certainly, I'm sure, have a different understanding of what variables are important to measure, as well as specific distances and specific traits to narrow down on. And so we did our best to base these based on the previous information we had regarding barn owls in a system very close by in Napa with lots of our previous data, as well as looking at just qualitatively some of the owls' tracks and hearing about some of the stories of how other folks have observed the behavior of these owls in our system. But it is truly subjective, and we definitely...
Oh, well, don't be so harsh with yourself. I think it's not purely subjective. I guess we had some measures, but I guess that's so interesting that you could show that they are repeatable. So I think, no, let's say it differently. It will be interesting if you have a new set of data and you would come up with the same solutions on which behavior falls into the same kind of PCA structure. I find this so interesting, and I'm really admiring that you have done that, because that's not it's not easy to do. So I really think it's amazing. Yeah.
Thank you so much, Christine. I'd love to talk more with you about this, but for time's sake, we're gonna go ahead and take one last question, maybe, or if there's a couple more.
Awesome presentation, really great work. I've seen that the plot a couple times before, some of your other presentations, all the different owls and how much they use vineyards, and I always find it so shocking that some of them use almost 100% vineyard and some of them didn't use it at all. Do you think that sort of variation, would you hypothesize that sort of variation, and such a what appears to my layman eyes, such strong variation, do you think that would apply to other systems as well? Like outside of the vineyard landscape, or do you think that these individual differences are really coming to light because of the system, or like, yeah, or would that sort of carry over to other systems?
In barn owls specifically?
Yeah.
Yeah, great question. This is definitely something I've been thinking a lot about recently, and I think it kind of comes down to like there's this inherent difference in what the landscape looks like around each nest. And so what drives each individual's actual selection and use of different landscapes could be based on some decision that they make much earlier on in terms of like, why are they even occupying this nest in the first place? Is it because of the habitat around it? And a lot of Matt's other research has shown that barn owls do select nests depending on the broader local landscape. But the motivation for individuals to select different habitats is really interesting. And one of the key concepts behind this study and including this movement strategy and neophobia designed together is that these traits have been found to be really linked in a lot of other taxa. So animals that have a bolder tendency or are generally more aggressive or risk-taking or exploratory can use landscapes differently. And each of these landscapes is structured, or each of these land cover types is structured differently. So there might just be a more inherent risk to using one versus the other. It depends a lot on the circumstances of the actual location, but it's interesting to think too, like what the actual motivation of the owl is to use or even occupy those landscapes in the first place. So yeah, I hope that answered your question.
Totally. Thank you. Yeah, I'm just curious for your thoughts on that type.
Yeah, it's a puzzle for sure. I wish that I knew everything about it, but yeah, thank you. One more question.
Always one on chat.
Oh, let's see, we've got chats. I don't know if there's a question. Yeah,
I
right.
would double dip if anyone else has a question. So again, it's fascinating, especially thinking about predators, in an individual behavioral syndrome context. I think about, behavioral flexibility, something that is a fairly top level predator having a lot of cognitive flexibility and the ability to plastically change behaviors. And I think one of the biggest things that tends to come into play for that is things like state dependent responses. If you're a relatively fat individual, socially dominant, whatever the case may be, adaptively changing your behavior and landscape use over fairly short times, can really complicate trying to assess repeatability of behavior. And so I remember when you guys captured your owls, you mentioned taking a lot of morphological characteristics. You end up assessing mass and, with acknowledgement to the complications and confounding things that can affect that. Do you think individual mass or individual state might have had a factor or a role?
Yeah, that's a great question. We haven't looked at that quite yet, but you're absolutely right. We have a lot of the tools at our disposal to look into that in the future potentially. We did collect measurements on all of the body morphometrics, so like tarsus, wing, Coleman, the whole 9 yards, along with body mass, both on initial deployment and on retrieval. So we have some information about each individual owl's size and also potentially body condition. Which could be interesting to look at with some of these factors for sure. I think there could be potential links there, but your guess is as good as mine at the moment. I don't know. The fat owls might stick a little closer to home. All right.
Okay, yeah, I think with that, we're gonna close, but where's the closed door session? Are we doing it here? Are we going down to 142? Okay, we'll go down to 142 with Barbara and Tim.
We'll meet you on Zoom.
We'll meet you on Zoom down there. So we'll sign off for now. Thanks everybody on Zoom.